All right, we're going to introduce the topic of DNA. You guys have already read 2 sections about it. So we're going to go over the structure of DNA. We're also going to go over and discuss the individuals who contributed to our knowledge about DNA. The scientists that we're going to go over, they all were trying to figure out what was the source of the genetic material. And through their experimentations, they found out that it was DNA and not other things. So our current knowledge of DNA is only about 90 years old. We learn more and more information each year about DNA. First discovered in 1928 or around that time. And then it says that they were able to actually map all of our genes. For example, in humans, we have about 30,000 genes that they figured out in the early 2000s doing something called the Human Genome Project. And again, like I mentioned before, there are tons of new discoveries being discovered all the time about DNA. So the first scientist that we're going to talk about is Frederick Griffith. And what Frederick Griffith did was he studied pneumonia. Pneumonia is caused by bacteria. That is why when you have pneumonia, they give you some sort of antibiotics along with other things, because the antibiotics work with bacteria, but they do not work with viruses. So one of these forms of bacteria is harmless. The other one is harmful. So the pathogenic, anything that causes disease is termed pathogenic, and anything that does not cause disease or is harmless is non-pathogenic. So he injected mice with the harmless form. Nothing happened to the mice. He injected the harmful form, and obviously the mice died. He then injected a combination of the harmless and the harmful bacteria, and he noticed that the bacteria died. He was wondering why. Well, bacteria have a process called transformation. They can take up the genes or the genetics of another bacterium and make it part of their own. So the harmless bacteria took up the DNA of the harmful bacteria, and that's why when they were together, the mouse died. Okay. This is just pictures of what we were talking about there. You have similar pictures in your textbook showing us the different situations about injecting the mice with harmless and the harmful forms, and then the transformation process, which is illustrated right here. All right, so the process by which bacteria are able to take up the genetics of other bacteria and implement it into their own is called transformation. Now, the factors that they're talking about are genes or traits or whatever it is. All right, so Oswald Avery. Oswald Avery is another scientist who piggybacked on the other experiment with dealing with mice, okay? The transformation process. So Oswald Avery also worked with heat-killed bacteria. And again, they were trying to figure out what was the transforming principle. Was it a protein? Was it carbohydrates? None of that caused a change in the organisms like nucleic acids did. Okay, so it was the nucleic acids, DNA in particular, that were passed on and caused a change in the transformation principle. All right, Hershey and Chase, what Hershey and Chase did is that they discovered that if they use radioactive material, now the radioactive material is important because they can actually use this to trace through an organism, through the body of an organism, maybe where the item is that they're looking for. So what they did was that they used radioactive phosphorus to find the DNA and radioactive sulfur to find the protein. And when they allowed these bacteria to reproduce, the bacteriophages, which are viruses that infect bacteria, when they allowed the bacteriophage to reproduce, they were going to see what was passed on to the new organisms. Well, what was passed on is the DNA. Okay, the radioactive DNA was passed on to the new organism and not the phosphorus. Thus, supported their principle that DNA is the source of the genetic information. So these bacteriophages, they look like either an alien spaceship or some kind of alien. Now, here is the phosphorus, which is found in DNA. Okay, so as we see, when the DNA was injected inside, this was what was passed on. The protein coat was not passed on. Again, that supported their experiment and suggested that DNA is the source of the genetic information, not the protein coat. We know that from previous lectures, we know that all of our heritable information, the stuff that we pass on, is found in our genes. We also know that through the process of mitosis and meiosis that our genetic information is copied fairly easily. It's not a very detailed process. And we know that these characteristics, these heritable characteristics, are passed on from parent to offspring along generations. So there are three parts to the DNA molecule, okay? Our DNA's main part, building block, is a nucleotide. A nucleotide has three parts. It has a 5-carbon sugar, has a phosphorus group, or phosphate group, which is made of phosphorus, and then it has one of 4 nitrogenous bases. Adenine, guanine, cytosine, or thymine are the 4 bases that we find in DNA. So here are the 4 different bases structurally. The 2 smaller ones are cytosine and thymine. They're called the pyrimidines. We can tell that they're smaller because they only have 1 ring that represents their structure, whereby the purines are double. They're larger. They have 2 rings that represent their structure. All right, guys, so our 4 different bases here, cytosine, thymine, obviously are the smaller ones. How do we know that? Because they are single ring structures. They're called pyrimidines. And adenine and guanine are the larger ones. They are called purines. And we can recognize a purine by looking at the double structure there. This Erwin Chargaff. What he came up with is called Chargaff's rules. So we have 4 different organisms here. Now what you're looking at is look at the approximate amounts of adenine compared to thymine, and then guanine with cytosine. They are not equal, they are approximately equal. So Streptococcus has about 29.8% of adenine and 31.6% of thymine. That is not equal, but it's approximately the same. So, all right, now this scientist is the individual who discovered the process of X-ray diffraction. She also discovered that there were 2 forms of DNA, an A form and a B form. Now, interestingly, she is the reason why Watson and Crick were able to build their model. They finessed her model. No, yes, they did. They stole her picture, her X-ray picture, and they used that information without her knowledge to build their model. Now, ultimately, what they got was that they got a Nobel Prize from building this model. Rosalind Franklin, due to some complications, she got cancer from probably dealing with a lot of the X-ray information. She died. She did not get any Nobel Prize whatsoever. They get most of the credit, yes, and it was because she was a woman, and at that time, women were discriminated against. And the other thing is that when they were working in the labs and stuff together, her lab office was down in the basement. Their lab office was up on the floor somewhere. So it was not a very hospitable place for a woman scientist to be in. Here's a picture of her X-ray diffraction photo. And again, this is the information. Now, we look at it like, oh, okay, well, we can't really tell. We see that some X's here, but it's actually a double helix. Is a double helix, and people who study X-rays are able to see that that is a double helix. So Watson and Crick were able to take this information, and they were able to build their model. All right, so here are the some pictures of these individuals. We have Crick, Watson, who's an American, Maurice Wilkins, he was the director of the lab that they all worked in, and there's Rosalind Franklin. Now it says that they made cardboard cutouts of the nucleotides. Well, they did not know how to place these nucleotides and what order to place them until, again, they saw her X-ray diffraction photo. Then it was an aha moment, and they were able to figure out that DNA runs from 3' to 5' on one end and 5' to 3' on the other, which means that it's anti-parallel or it runs upside down. Picture of Watson and Crick. and this is the model that they made out of metal parts and things like that. But they are very famous for creating this model, which gives us a basic understanding of how DNA looks and works and operates. I told you that DNA runs in opposite directions. So one side is going from top to bottom, and then the other side is going from bottom to top. Now the 5' represents where the carbon is. So that's the 5th carbon. 1, 2, 3, 4, 5. And the 3' right here is the 3rd carbon. So that's 1, 2, 3. All right, so we have to recall and remember talking about how the letters complement one another, or base pairing in DNA. Adenine complements thymine. It goes in the opposite direction as well. So thymine is going to complement adenine. Guanine complements cytosine, and cytosine complements guanine. Now, it says equal there, but that is, it can be misunderstood because they are not equal in amounts. Remember, Chargaff's rule states that they are in approximate amounts, not totally equal. And they are bonded together by hydrogen bonds. So we can look here. This is adenine. Adenine is a larger double ring structure. Thymine is a single ring structure, so we can always identify them based on that. Guanine is double ringed, and cytosine is double ringed. All right, so we're looking at prokaryotic DNA, and we're going to differentiate between this DNA and the DNA of the eukaryotic. This blue picture here with the yellow chromosome shows us the circular chromosome of the bacteria. Notice that it is smaller in length. Also, bacteria have less base pairs than eukaryotic organisms. The other picture we see, the green picture with the orange bacteria, just shows that a bacterium that was broken and its DNA is leaking out. Looking at the eukaryotic DNA information, if we compare the two, prokaryotic with eukaryotic, we see that the DNA is listed on many chromosomes as opposed to just circular loop of DNA. We also know that variation exists among species for the eukaryotic DNA. Look how it's 1,000 times more than that of prokaryotes. It's also larger in diameter and length. There are also different structures that you find in eukaryotes that you don't find in prokaryotes, such as histones, nucleosomes, and chromatin. These are all different structures that help our DNA be condensed and compacted to fit on the chromosomes that we have. Now, if we're looking at this diagram, we would want to look at it and review it with our notes going from right to left, because starting on the right we have our double helix of DNA, and it's unwinding to the components that make it up. So we have the histones, and notice how the DNA is wrapped around that tightly, and then it becomes nucleosomes. We have the coils all present there, and then we see the supercoils. This is how our DNA gets wrapped around and compacted so that it fits on the chromosomes that we have. Okay, those 30,000 genes and stuff like that that we have been known to have had using the Human Genome Project as a reference, approximately 30,000 or so. DNA has to copy itself, so that's why we're going to have to go over replication, explaining to us how DNA copies itself. We already went over the basis of this, and we know that DNA copies itself in S phase of interphase. Okay, that's when the DNA is duplicated. Why? Because we want each one of our cells to have the same DNA information, and this is how it happens. So if we have one side of our DNA that has strands adenine, adenine, guanine, cytosine, thymine, thymine, and thymine, what will the new match be? So we're trying to find the complement. So that blank that exists in the notes, you have to add the complement to this strand of DNA. Prokaryotes, guys, replication in prokaryotes is very simple. You have structures like these loops here that are present. 2 new loops are made. They have a cell wall, a plasma membrane. Okay, that's not especially unique. However, the forming of these loops is. The other thing that's unique too is the fact that you have a septum forming here before the two cells separate. In eukaryotic cell division, you have a cleavage furrow forming before the cells divide. So that is a different process that happens, and a very simplified process compared to the meiotic and mitotic divisions that happen with eukaryotes. So replication in eukaryotes, as we know, is much more complicated. And again, the reason why the DNA has to replicate or duplicate or double itself is so that the same genetic information is copied and passed on to the new cells. Okay? And in this instance, the same DNA is copied so that it is passed on to the new organism. And when I mean organism, I mean new cell, new DNA, which has a template of the old strand of DNA. So, template also can be worded as a model or an example. So each new strand of DNA is a combination of the old strand and a new strand. Enzymes, very helpful structures, proteins, as we know, unzip the information, unzip the DNA molecule, allowing for the new base pairs to be added to make the new strands of DNA. It also proofreads it. Okay, so it makes sure that any kind of corrections that can be done are made. All right, so guys, number 1 represents the original strand of DNA. Number 2 illustrates when an enzyme like DNA polymerase comes along, splits the DNA in half, or unzips it is the terminology that is used. And notice those new base pairs are going to be added to the unzipped strand of DNA. So from that one original strand, now in number 3, we have 2 strands of DNA that have both old and new parts to it. This is the more realistic diagram, as it says at the top, because DNA looks like a spiral staircase or a twisted ladder in actuality. And so this shows us what it could potentially look like. It also color codes the adenines, guanines, cytosines, and thymines. Shows us that polymerase operates at more than one point, which is what actually happens. Shows us the original strand and the new strand of DNA. All right, guys, we're going to go over the reason why we have, or one of the reasons why we have DNA molecules present. And one of the things that we can do from that is we make proteins. Why protein creation is important? Because all of the chemical reactions that occur in our bodies, all of the structural components, whether it's because of how your blood flows properly without issue, your body temperature operates automatically without you thinking about it, all of that stuff is due, the shape of your eyes, the hair color, your skin tone, all of that stuff is due to proteins being created from the DNA in our cells. Okay, so these notes just detail some review information about DNA. DNA has the gene information that has to be copied by RNA, and we're going to go into details about which RNA does that. DNA stays in the nucleus. It's kind of like the blueprint of the cell. It is analogous to an architect building at a job site and having a blueprint, but if you are an architect, you don't take your master copy to the job site, you take notes, okay, or you take a copy of it, all right? And that's kind of what happens with DNA. The genetic information for us has to stay in our cells, so therefore there has to be a way for that information to get out and to make proteins. This happens at the ribosome. We talked about that before as the organelle. We also find it on the endoplasmic reticulum. And again, DNA is unable to leave this nucleus, but the information in DNA has to leave the nucleus so that protein synthesis can be carried out. Okay, this compares and contrasts RNA with DNA. So tomorrow on the potential quiz that you will have, you're going to have to know the differences between these molecules. Number 1, it's single-stranded. DNA is double-stranded. Number 2, there's a different kind of sugar. We have ribose instead of deoxyribose. Thirdly, another difference is that we have uracil instead of thymine. We also have RNA polymerase that works with RNA, whereas DNA polymerase works with DNA. And this can leave the nucleus. Remember, DNA cannot leave the nucleus. So you have one side copy of the DNA molecule that leaves the nucleus. And the reason being, again, as we said when I introduced the topic, is to make proteins. Very important to keep going back to why protein creation and regulation is important. Okay, so 3 types of RNA. mRNA, also called messenger RNA, its job is to help unzip the DNA strand, and then along with RNA polymerase, that enzyme, it helps messenger RNA copy the DNA's information. Then what's going to happen is that it's going to leave the nucleus out of a nuclear pore. It's going to go to the ribosomes. There are 2 types of ribosomes. You have a small subunit and a larger subunit. And at the ribosomes is where protein synthesis occurs. So working with transfer RNA, ribosomal RNA, and messenger RNA, this is how proteins are created in organisms. Okay, there are 2 types of things, or 2 things that happen in protein synthesis. Transcription and translation. And the 2 of them together is called gene expression. Genes expressing themselves. Your eye color, your skin tone, your height. Those are all physical phenotype expressions of what's in our DNA. Okay, just like the heading states, we are transcribing DNA language into RNA language or protein language. RNA polymerase assists us here, as I explained before, helps unzip the DNA molecule. Then that messenger RNA comes along, matches one strand it matches one side of the DNA molecule. It begins at a place called a promoter. It concludes at what's called a terminator. And you look at these words and they kind of tell you what happens. Now, introns are kind of like blank pieces of DNA. They don't code for anything. There are no letters there, but they're not harmful either. Exons have letters or information. So what's going to happen is that these non-information pieces are going to be cut out. Kind of like a rough draft where you correct your mistakes and you take out the stuff that you don't need. Maybe extra paragraph here, extra sentence there, you take it out. It's not needed. All right? What you have is your final copy of messenger RNA. So exons code for information, introns do not. It is the completed RNA strand that's going to leave the nucleus, not the ones with the introns in it. Okay, this diagram, you'll see it again when you do your reading, vocab, and assessment tonight. Color codes the base pairs. It also shows us which ones happen in DNA and which ones happen in RNA. So we know that adenine both occurs in RNA and DNA. Cytosine occurs in both. Guanine occurs in both. But look at thymine. It says DNA only. Uracil, DNA only. That yellow structure there, that is a nucleolus. Okay, the bigger purple one is the actual nucleus. The holes in the nuclear membrane are the nuclear pores. That's where the messenger RNA gets out. Okay, if I didn't even put labels on this diagram, this should show you, you should say, oh, okay, that's a single-stranded molecule. Which one is RNA, which one is DNA? You would choose that one for RNA. Then you say, oh, okay, easy question. Which one is a double helix and represents DNA? And we see that there. Okay, so you guys got to pay attention to the diagrams. They have a lot of information in them. Again, the heading but it does tells us what happens during this process. So the messenger RNA information is going to be translated into a protein language so that amino acids can come together. Why? Because amino acids coming together make proteins. Why is that important? We hammered it out before. proteins kind of control everything that is us. Okay, every reaction, our physical features, any chemical reaction that occurs in our bodies, enzymes, speeding things up, those are all proteins. So there are 64 different combinations you can make. And we're going to go over that 4x4. And then there are 20 amino acids. Those 20 amino acids can arrange themselves into several different proteins. 3 letters, adenine, uracil, and guanine, will stop the process. And we also have stop codons. Now, you may say, well, why do you want to stop production of something so important as making proteins? Well, our body's about homeostasis, balance. Okay, you're only going to make what you need. And then if you don't need it, you store it or you get rid of it, right? So protein creation, your body may only need a certain amount of proteins, and that's it. All right, there's a part missing here. So what I want you to do is you're going to take a picture of this chart, and then I'm going to tell you to add another column here. Okay, we're going to add a column to the right side, and I'm going to explain to you how to read this chart. All right, so what you need to draw is another column. And in that column, I want you to put uracil here, cytosine, adenine, and guanine. It needs to look just like this one on the left. So color-coded also. So this same 4 letters in a column format, you're going to copy that on this side, because we need our third letters. And this chart doesn't show you where to get your third letters. It shows you where to get your first one, your second one. You have to write the third one. So the way that I get my 3 letters, I start here on the left side, I go to the top, and then I'm going to go to the right side. 3 U's. The abbreviation for that amino acid is PHE. U, U, and C would give me PHE also. Phenanine. Okay? U, U, and A, leucine. U, U, and G, the G is way over there. Not this G, the G on the side that I asked you to draw. Okay? Or actually, it is that G, I'm sorry. All right, so 3 different letters help make the amino acids. All right, so peptide bonds connect amino acids. Jot that down, please.
Wait, what?
Peptide bonds connect amino acids. Peptide bonds connect amino acids. A polypeptide is a long chain of amino acids. A long chain of amino acids will make protein. So the anticodon that you see there is associated and attached to the transfer RNA. The codon is attached to the messenger RNA. So if I say that anticodon and codon complement one another, I am referring to the same thing that happens with DNA. So in DNA, just as an example, adenine complements thymine, guanine complements cytosine. In RNA, we know that adenine complements uracil, guanine complements cytosine. So the letters are going to complement one another or match or pair up. All right, so this diagram is similar, if not the same, as the one that's in your textbook. Again, let's look at the structure here. We have our messenger RNA molecule leaving the nuclear pore, single-stranded. We have our large subunit ribosome, smaller subunit ribosome. So this protein synthesis, this creation of this polypeptide is happening here at the ribosomes. So what will happen is that a transfer RNA molecule with an amino acid attached will keep coming here till it reaches the stop codon. It's going to drop off its amino acid. Polypeptide bonds are going to form. And ultimately what you'll have in the end is that you'll have a chain of protein. Okay? And this chain of protein could be anything. It could be eye color, height, hair color, whatever it is. When you get to your stop codon, you stop production for the reasons that I explained to you. Enough proteins are produced, the body doesn't need any more of that particular protein at the time, it cuts off production temporarily. All right, so I explained to you the products of protein synthesis. I gave you examples of why it's important. Well, they have a big eyeball there with a blue coloring, and the reason why they're using this as an example is because that is a product of protein creation. Okay, the color is a product of protein creation. So it says, after translation, the protein can be modified at the rough ER. We know that the difference between the rough ER and the smooth ER is the presence of ribosomes. The protein can also, or it can stay in the cell. That's the second bullet. Third bullet says, it can be packed by the Golgi body and shipped out. That's one of the jobs of the Golgi apparatus. Remember, it's kind of like UPS, DHL, or the Postal Service. Then it gives us this example here. It says, you have brown eyes. That means that you have the gene that codes for brown pigment to make your eyes appear brown. Okay, your DNA is copied and the pigment is made. So your DNA specifies what your eye color is going to be, and then it is expressed when the protein is made. Okay, certain mutations, as we're going to learn, these are chromosomal mutations and gene mutations. Now, to differentiate them, an error that happens for a single gene is not as detrimental or bad as an error that happens on an entire chromosome. Why? Because, for the obvious reasons, it's a numbers game. Chromosomes have many genes on them, and if you have an affected chromosome, you're affecting many genes. However, if one gene is missing, it might not, or probably is not as severe. So, we have one example of a mutation. It's called a substitution. It is also a point mutation. In this example, guanine was substituted for cytosine. That, in the end, is going to make a different set of 3 letters, which then is going to make a different amino acid. In this second example of an insertion or a deletion, they added an extra adenine to it. Again, when you look at those 3 letters, it's going to read differently than this message. Okay? That's called a frame shift mutation because it's shifting the message. It's shifting the letters. Okay? All right, so other mutations. Okay, other mutations are more chromosomal based. So when chromosomes separate, another terminology for that is disjunction. They separate properly. Non-disjunction means that during meiosis or mitosis, the chromosomes did not separate. During metaphase, they didn't go where they were supposed to go. They didn't go to the right, they didn't go to the left. Too many went here, too few went there. When that goes into your sperm or egg cells, there could be a problem. Now, Down syndrome is different. It's not in the sperm or egg cell, but it happens at chromosome number 21. Okay, Down syndrome happens at chromosome number 21, and the individuals have an extra chromosome. Now, all Down syndrome individuals, no matter what ethnicity, culture, race, whatever it is, they have the same kind of features. Because at chromosome 21, when there's an extra chromosome there, it creates those different features. Yesterday, I told you guys about Down syndrome. Now, Down syndrome, again, occurs at chromosome number 21. It is also called trisomy 21. And again, the reason that most Down syndrome individuals look the same is because all of these characteristics and features fall here. Okay, that's why no matter what ethnicity, race, place of origin, people with Down syndrome have some of the same physical features. Now, their cognitive abilities vary. Okay, you can be a little higher on the end or a little lower. That varies. All right, so we know that our N number is 23. That is the amount of chromosomes that you find in our sperm and egg cell, our sexually reproducing cells. Therefore, 23 plus 23 equals 46. So we have a 2N number, okay, unlike plants and some other organisms. This is very normal for plants, not in us. If we have extra chromosomes somewhere, there's a chance that there may be a problem. Okay, so pictures display various types of mutations. Picture here with the birds. Okay, these are variations, but they are slight differences or slight mutations from whatever the normal phenotype is. Not harmful at all. Okay, no different than the variations that happen in humans. Okay, skin color, ethnicity, all that stuff. Those are all variations. And really, race has very, very, very small genetic, you can't even really trace it in human beings, truthfully. All right, so we have polydactyly, okay, which means that you have an extra finger or an extra toe, okay? This, I don't remember the name of the disorder, but basically, I think it's nicknamed like werewolf syndrome or something like that. You just have excessive hair growth. Oh, why don't you shave it? Well, it comes back. Okay, it's kind of like there are some women who grow mustaches and facial hair, and you shave it and it still comes back. All right. Again, I don't know what the name of this disorder is either, but I do know that as he matured as an adult, that muscle definition went away.